Hello everyone, and today's topic to be discussed, that is digestion and absorption of lipids. Digestion of lipids is type of lipids are in various types, simple to complex, so that we have already learned in chemistry aspects of lipids like simple lipids, complex lipids and derived lipids. So here the food or the diet or typical Indian diet is a combination mix of all the complex and simple lipids, and how these simple and complex lipids are undergoing digestion, and what is the aspects of the absorption, transport, and how they are entering, how they are transporting the circulation, that all we'll see in this video. So the average normal Indian diet contains 20 to 30 grams of lipids, okay, per day. And when you compare our Indian diet with the Western diet, okay, their diet is more rich of cheese, butter and other ice creams, okay, and more dairy products, so which are rich of lipid content. So their diet will be consist 2 or 3 times more than the quantity of Indian diet. So coming to the basic aspects of digestion of lipids, okay, where it is taking place. Okay, firstly, if you see digestion of lipids, though there is no time for the diet that we consumed, okay, in the mouth, okay, the whatever the enzymes present in the mouth, they will get sufficient time to get digested. So the food content that we place in the mouth will sequentially transported to the stomach. So though there is a presence of enzyme like digestion of lipids in the mouth because of lingual lipase, but we are not giving much time. Okay, compared to the carbohydrate part, lipids will take time. Okay, more complex linkage breaking has to happen here. So the enzyme lingual lipase from the mouth enters into the stomach along with the food. Unlike to the salivary amylase, here salivary lipase or lingual lipase, okay, it has got optimal pH 2.55, so that means it can work in acidic environment. Okay, so this enzyme therefore continues to be active in the enzyme stomach, and it acts on short chain triglycerides. Short chain triglycerides in the sense glycerol will be there, okay, and to this fatty acids which are of short carbons like 5 carbons or 6 carbons, okay, or 8 carbons or below 10 carbons. So like these fatty acids will be attached. So they are known as short chain triglycerides. And short chain triglycerides mainly present in milk, butter and ghee, and the action of lingual lipase is observed more significant in newborn infants, because the milk, what it is containing, milk is containing of medium chain fatty acids and short chain fatty acids so attached to a glycerol. So for a newborn baby, the main diet is milk. Okay, apart from the lactose, these fatty acids are responsible for providing energy to the newborn baby. So that's why lingual lipase will play major role in digestion of these short chain triglycerides. And gastric lipase is also one of the lipase produced by parietal cells of the stomach, and it is an acid stable enzyme again with optimal pH about 5.4. It is secreted by chief cells and stimulated by the hormone gastrin, and up to 30% of digestion of triglycerides occurred in stomach. And physiological important lipases here in the table I am showing. Okay, one is lingual lipase, okay, that will be secreted in the mouth, but we are not giving much time in the mouth for the food to get digested. So mainly it will be, though it will reach us to the stomach, the main activity will be taking place in the stomach, and what it will do, it degrade the short chain triglycerides and converting them into free fatty acids and diacylglycerol. So like this, if this is the glycerol, 3 fatty acids are attached here. What will do, this lipase will transfer fatty acid, so now it will be with 2 fatty acids. This is called DAG, right? So this is the mechanism of action of lipase, lingual lipase, and pancreatic lipase plus co-lipase will be releasing from the pancreas, and its site of action is a small intestine. And what it will do, it will be acting on triacylglycerols with long chain fatty acids. So here it will be short chain, like short chain triglycerides. So the glycerol will be like attached with long chain fatty acids like 16 carbons or 18 carbons. So this is known as long chain fatty acids. They are known as long chain fatty acids. Okay, and here free fatty acids and 2 monoacylglycerol. Okay, the glycerol with 1 fatty acid. DAG means glycerol with 2 fatty acids. And again, intestinal is also able to produce some of the lipases. So these lipases will be acting in elongation with the bile acids, okay, and their site of action is small intestine. So these intestinal lipases breaking the THs with medium chain fatty acids. That means the carbons from 8 to 14 carbon atoms. Okay, and they produce more free fatty acids and glycerol. They completely hydrolyze the medium chain fatty acids. That means medium chain fatty acids, triacylglycerols into separate glycerol and free fatty acids. Okay, and phospholipase A2, which will be again site of action is small intestine. Okay, phospholipids with unsaturated fatty acids on position 2. What are the phospholipids? The phospholipids are coming under the category complex lipids. Okay, that means especially you can say phospholipids types are there. Sphingophospholipids are there, glycerophospholipids are there. So out of this, the nitrogen part will be separated out and free fatty acid will be separated. So unsaturated fatty acid and lysolecithin will be formed because of the action of phospholipase A2. And lipoprotein lipase is always present on the capillary walls. Whenever there is a triacylglycerol passing through, this lipoprotein lipase hydrolyzes the TAG into free fatty acids and glycerol. This especially seen in case of absolute need of energy. That means lipid degradation. And hormone sensitive lipase is working on adipocytes. So the main site of triacylglycerol store in our body is adipose tissue. Okay, where there is a need of energy, this hormone sensitive lipase activated in adipose tissue, it will start hydrolyzing the TAG to free fatty acids, the diacylglycerol and monoacylglycerol. So these are all the types of lipases right from the mouth, that is lingual lipase, pancreatic lipase from pancreas, intestinal lipase from intestine, phospholipase A2, it is also produced in the small intestine, and lipoprotein lipase is present in the capillary walls of the arteries, and hormone sensitive lipase present in adipocytes. All the target or the aim of these enzymes to produce free fatty acids and the derivative products of triacylglycerols. Okay, and next coming part is digestion in intestine. So the main thing is emulsification. So emulsification is nothing but there is absolute need, because lipids are insoluble in nature. Okay, at the same time our enzymes require more surface area to work on the substances. Being insoluble, these enzymes what are releasing from the intestine and from the pancreas will not be working better. So emulsification is nothing but mixing of the lipid food with the bile salts. Bile salts are here, glycocholic acid and taurocholic acid are the main bile salts secreted from the gallbladder along with the liver secretions. Okay, when they combine with this, they will form micelles. Micelles in the sense, they will dump all the insolubility of lipids inside and the soluble things will be on the outside. Suppose we have seen already lipid bilayer in the sense polar head and non-polar tail. So non-polar means water hating, okay, and polar head means water soluble. So all heads will be outside and tails will be packed inside. So they will make a round micelle type structure so that these enzymes can work better. So this is a typical micelle formation. So for making micelle formation, this emulsification process will take, it increases the surface area. Okay, so the lipids are dispersed in smaller droplets, surface tension is reduced, the surface area of droplet is increased. Okay, so that the efficiency of enzyme activity increases better with this emulsification process. So bile salts mainly getting this process emulsification by its detergent action, lowering the surface tension of the lipids to be immersed. Okay, and peristalsis, anyhow in the stomach, the peristalsis movement started from stomach to intestine and to the lower part of the intestine, continuous movement of the intestine. And phospholipids, they also have some sort of peristalsis movements, and the phospholipids are better for digestion in case of emulsification. Bile salts are important for digestion of lipids. Why? Because bile salts, they lower the surface tension, okay, and they emulsify the fat droplets, and the emulsification increases the surface area of particles for enhanced activity of the enzymes. You see here, fat will be like this, and bile salts, what they do, they lowering the surface tension, they will put all the polar heads outside and non-polar tails inside, so making a droplets type, and these droplets have more surface area to work on this. This is the surface area, so that enzyme will work better on these droplets, so they are efficient in breaking the droplets into smaller degradative parts. Okay, the hydrophobic portion of bile salt intercalate into the large aggregated lipid and the hydrophilic domains remaining at the surface. This leads to breakdown of large aggregates into smaller and smaller droplets. Thus the surface area of action of lipase is increased. Now pancreatic lipase in its action with co-lipase, so mainly as cholesterol esterase. Okay, when there is a combination, the cholesterol So this is cholesterol. So if there is any fatty acid attached here, so this is known as cholesterol ester. So for absorption, cholesterol ester is not a suitable one. So to that, there is a enzyme cholesterol esterase which separates the cholesterol, okay, and free fatty acid. Now these free fatty acid cholesterol are suitable for transportation into the intestinal cells. Okay, and phospholipase A2, okay, this is the enzyme which separates the complex lipids such as glycophospholipid or sphingophospholipid, and the bile entering the duodenum serves to neutralize acidic chyme from the stomach and provides the pH favorable to the action of pancreatic enzymes. So this table we have already seen here: lingual lipase and pancreatic lipase, intestinal lipase, phospholipase A2, okay, and then lipoprotein lipase and hormone sensitive lipase, where and all they present, and what are the substrate they will be acting, and what is the product at the end. So first we will see one by one. Here, what is the action of these enzymes on triacylglycerols? So for triacylglycerols, if triacylglycerols are there in our body, okay, these are not suitable for absorption. So they are supposed to be converted into free fatty acids and glycerol. So pancreatic lipase will work on triacylglycerol. So adding water molecule, it break the fatty acid, so that you see here this is ester linkage. Okay, so fatty acids attached to the glycerol by ester linkage, and water molecule and lipase, it will break the ester linkage by adding water molecule. So it's a sequential removal. So what is happening? There is a one fatty acid has been removed. So now it become diacylglycerol. Again it will become one more fatty acid. Okay. So it will become monoacylglycerol. So finally, what is happening? There is one more. This means the whatever the fatty acid is there in the second carbon will be shifted to the first carbon. Now one more fatty acid has been removed. So then it will become glycerol. So the action of lipase will give you, okay, it will give you 3 fatty acids plus 1 glycerol. Pancreatic lipase acting on a TAG will give you 3 fatty acid plus 1 glycerol. And the major end product of the digestion of TAG is 2 MAG and 1 MAG, glycerol and fatty acid. So 100% means monoacylglycerol or preferable one, 78%, okay, and 1 MAG is 6%, glycerol and fatty acids 14%. The digestion of TAG is partial, okay, only monoacylglycerol will be absorbed, okay, and the cholesterol ester may be hydrolyzed by cholesterol and fatty acid, okay, and the action of phospholipase A2 produces lysophospholipid and fatty acids. Then only these formed products are suitable for absorption in the intestine. 𝐒𝐫𝐢𝐚. So what is 𝐒𝐶lipase? The binding of 𝐒𝐶lipase to the triacylglycerol molecule at the oil-water interface is obligatory for action of lipase. Okay, so that's why pancreatic lipase always associated with 𝐒𝐶lipase. So before action of the pancreatic lipase, 𝐒𝐶lipase has to come into the action. First it has to bind to the triacylglycerol. What to say, at the oil-water interface is a must happening, that efficiently pancreatic lipase has to work. The 𝐒𝐶lipase is secreted by pancreas as an inactive zymogen, okay, and is activated by trypsin. 𝐒𝐶lipase first initially it is secreted as inactive one, that is a zymogen, okay, and it is activated by trypsin. Next, coming to the absorption of lipids. So absorption of long chain fatty acids we'll see. So now digestion part we have seen. So triacylglycerols will be there, phospholipids are there, and cholesterol esters are there in the diet. So triacylglycerols will be converted to monoacylglycerols. Okay, sometimes monoacylglycerols plus fatty acid, okay, plus glycerol, okay, and phospholipids will be converted to lysophospholipid, plus fatty acid, and cholesterol will be converted to cholesterol plus free fatty acid, right? So these are the products. So now these formed products are suitable for absorption. Okay, long chain fatty acids means the chain length is more than 14 carbons are absorbed by lymph and not directly to the blood. So that's what lipid rich meal. Okay, our lymph is white in color because of the high concentration of long chain fatty acids in the lymph. Okay, the theory proposed by Breckstrom has the following steps. The bile salts, as I mentioned in the early side, the salt micelle incorporate products of digestion, namely 2 monoacylglycerols, long chain fatty acids and cholesterol, phospholipids and lysophospholipids into molecular aggregates to form mixed micelle. And this mixed micelle is easily transported in the circulation. These micelles are spherical particles with hydrophilic exterior and hydrophobic interior core. Due to their amphipathic nature, bile salts help to form micelle aggregates. You see here the typical representation of micelle. Here what happened, bile salts with their detergent actions. Okay, so cholesterol is there outside. Okay, monoacylglycerol is there. Okay, bile salt is giving a detergent action, and long chain fatty acids are packed inside. Okay, because fatty acids are insoluble, so they are packed inside. This is a perfect example for a micelle formation, and this is a mixed micelle. Cholesterol is there, monoacylglycerol is there, and fatty acids are packed inside. So this thing can easily be absorbed by the intestine. So absorption of fat as chylomicrons. So after absorption, for transportation from intestine to liver, there we need a carrier system that has been taken over by chylomicrons. Chylomicrons is a lipoprotein, that is combination of lipid plus protein, which carries the digested lipids. The process, it is also help of bile salts. You see fat droplet, emulsification is taking place in micelle formation. Then this micelle enter into the intestine, okay, by the process of enterocytosis and synthesis of, again, once it enter into the intestinal cells, the fatty acids combine with monoacylglycerol to form DAG and DAG to TAG, triacylglycerol. That means here before the intestine and once entering into the intestine, before intestine, to get into intestine they should be simple form. Once they enter inside the intestine, they should convert into complex form. That is the thing happening, and these triglycerols has been incorporated into lipoprotein, that is chylomicron, and they can easily be transported in the circulation from intestine to liver. So chylomicrons or the lipoproteins are acting as a carriers for exogenous triacylglycerols. That means the triacylglycerols that are coming from the diet will be transported by chylomicrons. So micellar formation is essential for absorption of fat soluble vitamins such as vitamin A, D and K, and micelles are aligned at microvillous surface of the jejunal mucosa. Fatty acids, 2-MAG and other digested products passively diffuse into the mucosal cells. So they don't need to form any micelle formation and all. So fatty acid 2-MAG can easily be enter into the intestinal mucosal cells. So absorption, so that's what we said. Long chain fatty acids are absorbed into the intestinal cell wall where they are reesterified, incorporated into the chylomicrons and enter into lymphatics. Short chain medium chain fatty acids are directly absorbed into the blood capillaries. Bile acids are reabsorbed into portal vein. So enterohepatic circulation of bile salts, the bile salts are left behind, which are mostly reabsorbed from the ileum and returned to the liver to be re-excreted. That is called enterohepatic circulation. About 98% of dietary lipids are normally absorbed without any aid. So this is the main step before breaking of ester linkage, and once they enter into the mucosal cells, reesterification has to take place. Once they are inside the intestinal mucosal cell, long chain fatty acids are reactivated from triacylglycerols, and the fatty acids are first activated as fatty acyl-CoA by the enzyme acyl-CoA synthase or thiokinase. Thiokinase is mainly present in intestinal mucosal cell, and this need lysis of two high energy bonds. That means you have to spend energy in mean of ATP. Two such activated fatty acids react with monoacylglycerol to form triacylglycerol, and majority of the molecules follow this MAG pathway. MAG in the sense monoacylglycerol pathway. Free glycerol absorbed from intestinal lumen directly enters into bloodstream, and there it go to adipose side, and where they will be accepting the fatty acids to form TAG. So free glycerol is not available for reesterification, but the cells can derive glycerol phosphate from glucose by glycolysis and add 3 molecules of acyl groups to synthesize TAG in adipocytes. So chylomicrons, this what we have discussed, chylomicrons are the main lipid carriers from intestine to liver, especially for triacylglycerol. Okay, and cholesterol ester and phospholipid molecules along with apoproteins B48 and ApoA. So these are the characteristic proteins restricted to chylomicrons. Apoprotein B48 and ApoA are the proteins restricted to chylomicron. Because there are other lipoproteins, they are structurally different from the chylomicron in aspect of the proteins like apoproteins and other stuff. The chyle, milky fluid from the intestinal mucosal cells loaded with chylomicrons are transported through lacteals into the thoracic duct and then emptied into the systemic circulation. But the serum may appear milky after a high fat meal due to the presence of chylomicrons in the circulation. Normally the lipemia clears within few hours by the uptake of chylomicrons by the tissues. This is not a permanent one. Once they enter into the lymph, we can say, what is say, it's a systemic circulation, it will take time. Maybe it will take a few hours, maybe 4 to 5 hours. This will be cleared from the systemic circulation. So summary, you can say here, first bile salts, they are having digestive effect. Okay, in the stomach, there is a use of gastric lipase and the enzyme that is coming from mouth, that is lingual lipase. Both will work on the stomach, and then pancreas will release pancreatic lipase along with the 𝐾-lipase. It will work on it, and with the help of bile salts, emulsifying agents they will immerse the lipid droplets, and they have emulsification effect, and so that pancreatic enzymes work better on this, and THs will be converted into monoacylglycerols, free fatty acids, and then again intestinal lipase which will be acting on THs to form free fatty acids, and phospholipases will work on phospholipids to form isophospholipids and then free fatty acids. Again, we have one more, that is cholesterol esterase, which work on cholesterol esters to form free fatty acids and cholesterol. So finally, they will be entering to the portal circulation and to the liver, where from there they will be transported to the adipose tissue. Okay, the free glycerol will be transported to the adipose tissue from systemic circulation. It will be stored as triacylglycerol. When we need energy, that time these triacylglycerols will be broken down to glycerol and free fatty acids. Free fatty acids undergo beta oxidation to form ATP, and glycerol will be transported to liver, involved in gluconeogenesis process to provide glucose for production of energy. So 6 steps of lipid absorption: minor digestion of triacylglycerols in mouth and stomach by lingual lipase, major digestion of all lipids in the lumen of duodenum, jejunum by pancreatic lipase and lipolytic enzymes, and bile acid facilitates formation of mixed micelles for easing the absorption of these lipids into intestinal cells. Passive absorption of products of lipolysis form the mixed micelles into intestinal epithelial cell. Reesterification of 2 monoacylglycerol with free fatty acids and inside the intestinal enterocytes. Assembly of chylomicrons containing ApoB48 and ApoA along with triacylglycerols, cholesterol esters and phospholipids export from intestinal cells to lymphatic system, and then from lymphatic system to systemic circulation to liver. So 7 steps of lipid digestion absorption. You can say short chain fatty acids is different, like major digestion of lipids in the duodenum by pancreatic lipolytic enzymes. Bile acids formation requires what they facilitate? They facilitate the mixed micelle formation. Okay, reesterification, passive absorption to avail the products of lipolysis to form mixed micelles, and they facilitates the entry of mixed micelles into intestinal cells. Reesterification of 2 monoacylglycerols with free fatty acids inside the intestinal erythrocyte. Assembly of chylomicrons containing ApoB48 and ApoA along with triacylglycerols and cholesterol, which will be entering into the lymphatic system. So absorption of short chain free fatty acid is different. They are not reesterified and they are absorbed directly into the bloodstream, and they will reach us to the adipose tissue where glycerol will be reesterified to form THG. Short chain fatty acid absorption is different because in newborn babies especially this enzyme facility is not available. So in that condition, the milk is containing more of short chain fatty acids and medium chain fatty acids. Short chain fatty acids seen in milk, butter, ghee, and medium chain fatty acids do not need reesterification. They can directly enter into blood vessels, then to portal vein, finally to liver, where they are immediately utilized for energy. Their absorption is rapid. They are better absorbed than long chain fatty acids. So that's all about lipid digestion and absorption. Thanks for watching. Thank you.